In this figure, we want to look at the variety of cell-cell signaling that is non-neural. Right? In neural signaling, we had neurotransmitters released across a synaptic cleft, very small interstitial space distance to target a postsynaptic cell and change its function. In endocrine physiology, the chemical signal is also released into the interstitial space, but not across a synaptic cleft. In this case, we have several different versions. Now, the first three are generally not called endocrine classically. Classical endocrine is a blood-borne chemical messenger. And so these first several, blocked here, would be for what we call local control or sometimes autoregulation, right? And that's because cells within a particular tissue are responding to the environment there and initiating a change in response to that stimulus without any external input. One example of that is the one at the top here. It's called autocrine. And so in autocrine regulation, a cell will release a chemical signal which actually expresses receptors. These little V-shaped guys are supposed to be receptors. But the cell will express receptors for the chemical that it releases. And so what this means essentially is that a cell is modifying its own activity. In paracrine, we have cells releasing a chemical signal, again diffusing through the interstitial space out here, but now acting on an adjacent cell of a different type. And that's referred to as paracrine regulation. This is really common in microvascular control, where the local tissue cells are telling the microvasculature to change its resistance, modifying blood flow. There's a lot of other examples, but that's just one good one of a paracrine regulation. Endocrine regulation is fairly uncommon, and in this case, the cell producing the chemical agent doesn't actually release it. It produces a chemical agent that binds intracellular receptors to modify that cell's function. So again, some peptide protein hormones can act both inside the cell and be released, and their internal regulation is referred to as endocrine. Now, we get down here to sort of more classical endocrine and neuroendocrine. We have really two different versions. We have a version where we have classical endocrine tissues or endocrine glands that release their hormone into the interstitium. It diffuses into the bloodstream and then is carried by blood flow back to the heart and then into the general circulation, where it can leave that circulation at the majority of capillary beds. Again, randomly by diffusion, those hormones will find cells that express receptors, and only those cells that express receptors, binding those receptors. Sometimes they're found inside the cell, depending upon the chemical nature of this hormone. Here they're shown on the cell membrane, to initiate changes in the functioning of those target cells. Now that's classical endocrine, a blood-borne chemical messenger released by an endocrine cell. In neuroendocrine, we have a sort of an odd scenario where a neuron releases a chemical signal, which we would normally call a neurotransmitter, but in this case, releases it into the bloodstream, where it can then travel just as a regular hormone does and bind to distant target cells and modify their functions. And so really, neuroendocrine is often called endocrine. It is a blood-borne chemical messenger, but sometimes we're more specific in pointing out that it is being secreted from neurons. And there's only a few examples of this, the posterior pituitary being one of them.